Can someone frozen 50 years ago open their eyes today? In 1967, psychologist James Bedford became the first human to be cryopreserved. Today, at least 400 people worldwide lie dormant in liquid nitrogen at -196 degrees Celsius. Institutions like Alcor receive new cryopreserved individuals every year. They are betting on the future, but what are the odds of winning this bet? Currently, no cryopreserved individual has been successfully revived. But this doesn't mean there's no hope. First, we need to understand what makes cryopreserving humans so difficult. The biggest enemy is ice crystals. Just like water pipes bursting in winter, water inside cells expands by 9% when frozen. Sharp ice crystals tear through cell membranes and destroy mitochondria. Once these powerhouses of the cell are damaged, life can't be restarted. To solve this problem, scientists invented vitrification, dehydrating cells with high-concentration cryoprotectants, then cooling them at 200 degrees Celsius per minute to turn cells into an ice-free glassy state. But a new problem arises: the dimethyl sulfoxide DMSO in the cryoprotectant is toxic, killing 10 to 20% of cells. Even worse is thawing. Even if freezing goes well, water rushes into cells frantically during rewarming, bursting them like overinflated balloons. That's why frozen strawberries turn mushy after thawing, and damage to human cells is fatal. In 2024, research from Fudan University in China broke this deadlock. The team led by Xiao Zhiqing developed a chemical mixture called MEATY. They cryopreserved human brain organoids for 18 months. Then thawed them. These mini brains not only survived but continued to grow. The neurons could even transmit signals. Professor Magalhaes from the University of Birmingham in the UK called it a miracle. Brain cells are extremely fragile. Surviving cryopreservation is already a breakthrough. Even more amazing, 46,000-year-old nematodes from Siberian permafrost, after thawing, could crawl, feed, and even reproduce. This proves that low-temperature dormancy can truly pause the clock of life. The field of assisted reproduction has long proven the potential of vitrification. Thousands of frozen babies worldwide have been born from thawed oocytes or embryos. But the gap between a single cell, an organ, and a complete human body is vast. The biggest challenge is organ complexity. A 2023 study showed that even small pig kidneys had 30% of cells die after cryopreservation and thawing. The human brain has 86 billion neurons, with connections more complex than cosmic galaxies. Any tiny damage could lead to permanent loss of memory or consciousness. The toxicity of cryoprotectants also remains unsolved. The most effective formula currently contains 15% DMSO, a chemical that damages cell membrane proteins and disables normal cell function. Not to mention the coordinated revival of the entire body. How to restart the heart? How to restore blood circulation? No one has answers to these questions. Current cryopreserved individuals are more like biological specimens. James Bedford has been frozen for 58 years. His cells may have already been damaged by the combined effects of ice crystals and toxins. Even if future technology exists, it may be useless. But research never stops. The Chinese Academy of Sciences is testing biomimetic ice control materials that can prevent ice crystal growth during thawing. The DMSO-free cryoprotectant developed by Songshan Lake Laboratory has increased stem cell revival rate to over 90%. An even more ambitious idea is nanorobots. Future ones may act like tiny doctors, repairing damaged cells one by one during thawing. Professor Roman Ball from the University of Surrey believes these technologies can only be used for medical research in the short term, such as preserving organs for transplantation. But no one can predict the future. 50 years ago, no one believed mobile phones could make video calls. Today, we can't rule out the possibility of reviving cryopreserved individuals in 50 years. Currently, cryopreserving humans is still a big gamble, but behind it is humanity's desire to extend life. If the technology matures, would you choose to sleep until the future?